It's my great pleasure to introduce Dr. Francesco D'Amico. Dr. D'Amico association with Elettra Sincrotrone Trieste commenced in 2008 when he joined as a postdoctoral researcher, collaborating with Dr. Claudio Masciovicchio group. His contributions extended to the design and construction of the TMX and the station. Aimed at facilitating experiments in the UV band to investigate the hydration properties of the organic molecules. Between 2012 and 2013, his research was dedicated to the design and the construction of a high-resolution ultraviolet Raman spectrometer based on synchrotron radiation. At the Elettra IUVS Beamline since 2015, Dr. D'Amico has been actively engaged in biophysical applications of UVRR, particularly in the spectral analysis of nucleic acids and DNA. From 2018 onwards, he has broadened his research scope to include the potential use of UV source spectroscopy and investigations of biosystems with significant medical relevance. His research also encompasses the implementation of UV Raman techniques for the study of carbonation systems like graphene, graphite, and amorphous carbons. Additionally, he explores green materials based on tannins with intelligent properties. In his latest role as part of the Elettra 2 project, Dr. D'Amico is actively involved in designing and constructing the new IUVS 2 experimental station. His cutting-edge facility is dedicated to Raman measurements in the ultraviolet regime, utilizing a wide range of laser light sources. On behalf of all ROWS members, I welcome you to this webinar, and I also welcome all the invited guests and participants to the webinar. Now, I kindly request Dr. Francesco D'Amico to begin the presentation. Please.
Okay, so I can start. Basically, my talk will be devoted, as explained before, to explain the property of UV resonant Raman scattering, in particular to investigate what can be done with organic systems, in particular for biophysics, for the reason that we will explain during the talk, passing also to material science. Material science needs, for example, means in smart materials as tannin forms, for example, as flavonoids and similar materials, up to carbonaceous materials, graphene, graphites, as well passing to organic semiconductor, which is also an important category of materials that can be investigated in a very nice way with UV Raman scattering. A small fast introduction of vibrational spectroscopy in order to introduce in the best possible way the UV Raman scattering. For nonlinear molecule vibrates, we know, molecule vibrates, and we have from quantum mechanics a discrete number of normal modes of oscillation possible, each one with a specific vibrational energies, typically in the infrared range, or speaking in terms of energy, about something like fraction of electron volts, 100 of millielectron volts, more or less, or as typically used in spectroscopy, in wave numbers ranging from 10 to few thousand of wave numbers. And basically, from the vibration, we can collect in first properties of materials, and in second, we can deduce from variation in energies and in line shape of the spectrum, we can detect what occurs in the surrounding environment of the molecules. How we can investigate the vibration of molecules and materials in general? Basically, we have two techniques, infrared and Raman, that collect basically the same vibration, but with important differences that makes the two techniques strongly complementary. We have a sample that we would like to investigate. What we can do? We can send an incident radiation, polychromatic, in the energy range of infrared, so matching with the molecular vibration, and we can measure the transmission spectrum. What we found in the transmission spectrum. In the transmission spectrum, we found an intensity which is lower than the incident one, attenuated with the amount of the blue part of this graph, and this blue part of this graph is substantially the vibrational infrared spectrum. Each peak corresponds to a specific normal mode as seen before, and therefore from the position of this peak and from the line shape of this peak, we can deduce a certain number of properties. This is the first way to detect information about vibrations. There's a second way, which is based on scattering, and which is the Raman effect. In this case, the incident radiation is not in the infrared range, but is based typically in the ultraviolet or visible range. A monochromatic, in this case, incident radiation that is sent to the samples, and then what we collect is not the transmission, but the whole scattered light. The whole scattered light, how scattered light behaves. Scattered light, the most of the scattered light, 90.9% of photons, is elastically scattered with the same energy of the incident radiation. A small amount of this energy, actually, it is not elastically scattered, but inelastically scattered. Why? Because of interaction with the vibration seen before. The photons lose part of this energy to generate a vibration, molecular vibration, and it came out with a lower energy or with a higher energy in case a vibration is annihilated, given energy to the scattered light, and then we have these two parts of spectrum, which are not elastic, that are called Stokes and anti-Stokes spectrum, vibrational spectrum. These peaks are the same of this one, but with important difference that then we are going to investigate. This is spontaneous Raman. Then we talk also about resonance. In Raman scattering, as said before, we have 1 millionth of photon, more or less, sent, and order of magnitude or only one photon is elastically scattered. The same molecules as before. Here we have the vibrational Stokes spectrum. And in this case, we can recognize that each vibration corresponds to a specific peak. Here we have the sample of 3 peaks shown before, but basically all peaks have a specific energy. About the energy, what we have? About the energy, we have that here we are talking about Raman shift. That means in inverse centimeters. Basically, just to have an order of magnitude of the energy, we are talking about 1 milli electron volt corresponds to 8 inverse centimeters. It means that 0.2 electron volts are 1,600 inverse centimeters that correspond to the typical stretching in graphite sample. Just to have an idea of the order of magnitude, we have visible light and ultraviolet that are few electron volts. Instead, the scattered light is fraction of electron volts. In the classical model, just to have also the quantitative difference between infrared and Raman, what we have is that Raman bands arise from changes in molecular polarizability, differently from what occurs in the infrared, where variations are due to the dipole moment variations. And this has important consequence in the efficiency. Specifically, this means that the molecular polarizability determines the Raman cross-section, that the symmetric mode, like the stretching, like in this way, has a polarizability derivative equal to 0 and is Raman active. Otherwise, asymmetric modes have dipole moment derivative different to 0 and are infrared active. This difference is important because it means that the same samples may give a different same sample may give a different vibrational spectrum in infrared and Raman. What we can see immediately, just to provide you a real example of what I've explained up to now, is the spectrum of DNA. The blue one is the FTIR spectrum, the infrared one, the red one. The blue one instead is the ultraviolet resonant Raman. Raman spectra and infrared spectra are completely different, although they belong to the same samples. Basically, what we have is that this part of the molecules, which has high dipole moment, can be detected very well with infrared. Otherwise, the aromatic compounds coming from the nitrogenous bases instead is more has a more higher polarizability, and therefore their normal modes are better enhanced with Raman spectroscopy. And now we talk about resonant Raman. So up to now, we are talking only about spontaneous Raman. What does it mean, spontaneous Raman? Spontaneous Raman means that we are at the ground state, we have a vibrational excited state, and we have to find in this energy. We have to detect this energy. As we have explained before, we send a monochromatic radiation with a specific energy. Since Raman is a second order process, we need that we have a decay from a virtual state to the vibrational excited state in order to have the emission of the scattered light, which has a lower energy. And up to now, we are consistent and everything is okay. We have this virtual state and this works, and it provides that the probability of this event is a certain number. But what will occur in case of resonant Raman? In case of resonant Raman, we have to detect the same vibrational state, but in this case, the energy, the incident radiation is not at a casual energy, but as the specific energy corresponding to an electronic excited states. In other terms, we are moving really an electron from the ground state to an electronic excited state. This causes basically a modification in the chemical structure of the molecule we are analyzing. Basically, we have that the electronic environment changes basically stretching the molecules, and it reverses it into an increase of the probability to have the Raman scattering, because the polarizability increases a lot. And then we have this signal, this event that causes the emission of excited states, which is strongly increased in cross section. What does it mean? It means that in the case of a complex system in which we have a huge number of molecules, we have a complex system with different chemical species, what we have is that if in this case we have an strong increase of the resonant Raman cross section, if we have, for example, these two kinds of samples, one which is only spontaneous and one which acts as resonant Raman, we will see in the spectrum only the molecules belonging to the ones that have this effect. What does it mean, reversing in a real example about UV, and why UV resonant Raman scattering? Because here we have nucleotides, DGTP. We have guanine, we have the part of sugar, and this is guanosine, and this is the phosphate group. Three parts with three chemical differences, different in the chemical parts. This is the absorption spectrum of the GTP. It means that the most of electronic transitions are below 300 nanometers. Particular here we have two peaks in absorption, then we have a valley, and then we have a new increase in the absorption going to higher energies. What occurs? Let's see what occurs when we collect the Raman spectrum. If we collect the Raman spectrum, for example, with the excitation energy far from the absorption, so in spontaneous Raman condition, we have a certain number of peaks that belongs to normal modes of the molecules. Summarizing, we have this part that belongs to vibration coming from the nitrogenous species portion, and the yellow part instead that contains vibrations coming from the phosphate part. This in spontaneous Raman, but what occurs when we send an excitation inside this absorption peak? That means allowing an electronic transition in the system. In this case, what we have basically that we have an electronic transition from the HOMO state to the LUMO state of the molecule, more or less. We change the electronic state of only this portion of the molecule, not this one, and therefore the vibration coming from this side are enhanced. The line shape is slightly different because there are specific vibrations that are more enhanced than the other ones. And in particular, note that these two peaks belonging to this part are disappeared. And here we have instead different peak that belongs instead to this part that are enhanced. So in this portion, the spectrum is different, but not only in this portion. Let's see the higher part of the spectrum. Here we have the CH vibrations, basically, and here we have a tail that starts to rise due to the water contribution, because this spectra has been collected also in water environment. The same region instead, in the resonant spectrum, have a completely different line shape, which contain the same peak and further peak that rises, that are due to the overtones vibrations that are present, that are all overtones vibration could also be present in the spontaneous spectra, but are enhanced in when we have electronic transition and when we have resonant Raman effects, basically. So, summarizing, what we have is that if we have, in that case, we have a simple molecules. If we have a complex molecules with a UV visible absorption spectrum, with several peaks belonging to different molecular groups, I can ideally choose the energy, the excitation energy in this position, in this one, or in this one, to selectively enhance the molecular group 1, molecular group 2, or molecular group 3. This is an ideal case. In the real cases, for example, we have peptide bonds of proteins that have a strong absorption between 200 and 220 nanometer. It means that if we send an incident energy in this range, we can enhance the signal from peptide bonds. The pure world of aromatic compounds have different absorption spectrum. And for each time, we can select the energy to emphasize and to take attention to only one part of aromatic compounds with respect to the other one. The same is done for DNA nitrogenous bases and nitrogenous bases, because the absorption is in a wide region, and as we will explain better during the presentation, we can exploit the tunability in this range to investigate specific nitrogenous bases. The UV resonant Raman, apart from the enhancement of the Raman signal due to the resonance, possesses further advantages, which must be taken into considerations. The first one is that obviously we have the cross-section, the Raman cross-section that gives as the inverse of lambda elevated at 4, which is lambda is the wavelength of the excitation source. So in ultraviolet, we have a small lambda and we have a higher cross-section. And therefore, we should have a signal with a higher signal-to-noise ratio with respect to the visible one. There's a second important aspect. As the people that work with organic stuff or what, for example, archaeological samples on the other kind of samples, the visible range spectrum are always affected by strong fluorescence background, which is emitted together with the Raman signal, but since the probability of the fluorescence is strongly higher than the probability of the Raman scattering, often we have the visible spectra full of fluorescence. By moving to the ultraviolet, we can avoid this effect and we can have the spectra free from the aforementioned fluorescence background. Another important advantage is that it allows measurement in water and buffer solution, even at low solute concentration. So basically, this is the properties of Raman with respect to infrared. Also in spontaneous Raman, we can do measurements in water or aqueous solution that cannot be performed instead with infrared. But of course, with the effect of resonance, we have that this effect is more enhanced. For example, here we show several nucleotides measured at 532 nanometers, far from the resonance, in which we see that the spectrum, this is a 10 millimolar solution, is affected by the strong presence of a water peak that influences also the line shape of the spectrum. The same concentration, the spectrum done at 266 nanometers, so in the ultraviolet, in this case, we don't have this water peak, even if the concentration is the same, and we can appreciate better a flat background that allows to make a better considerations about the peak line shape. This is a strong advantage in the case we would like to compare the line shape, as we will done, as I will show you later, to make some consideration and data analysis. Another possibility is to use, for example, the exploiting synchronous radiation or also OPA laser to have a tunable CV radiation source in order to improve the selectivity, because in this case we have a laser that provides energy at 166 and we exploit only this. But if, for example, we have a better resonance 260 to 150, we can move to that to have a specific resonance. Just a brief reminder of what kind of setup we can have. We can have a standard macro setup in which we have basically a plano convex lens with the long focal length with a numerical aperture which is low more or less, and we have a classical collecting system with a broadband mirror with a plano convex lens that send the diffuser light to a spectrometer, and then the spectrometer analyze the radiation emitted. And then we can have, for example, the typical micro-Raman system that most of you probably knows, which is composed by a microscope objective, works with the beam splitters. In this case, we have a laser source, we have a first beam splitter, we have a second beam splitter that allows to the broad emitted light to send into the monochromator and allows measurements. What is the advantage of a measurement like this? The advantage is that since we have a plano convex length of a long focal length, we have a big spot size. We have a spot size something like 100 of microns. It is an advantage because, differently from what occurs in the visible range, the ultraviolet radiation could damage the sample. And therefore, the tricks to avoid the photoreal radiation are often used in UV resonant Raman, and we must care in this effect in order to avoid photodegradation of samples. Just a slide about monochromator, but you know. Let's talk instead about self-absorption of sample, which is strongly related to the photo damage of samples, possible photo damage of samples in ultraviolet. What we have basically, if we send an incident radiation to a sample, we have the emitted light that comes from a specific volume scattering. This volume scattering is a certain amount because typically light enters into the sample, but the attenuation increases while the beam enters into the sample, and also the diffused light that exits from samples is affected by this attenuation. And what occurs basically is that the diffused light in the X direction, the intensity of the diffused light is due to two effects, basically. This is when we have the penetration which is high, we have the whole volume which is covered. But in the case that we have the sample that strongly absorbs, we have only a thin layer which is sensitive to the Raman effect. So in this formula, we have two terms. We have terms that increases this term as the resonant Raman cross-section and the resonant sample concentration. More high is the sample concentration, more high is the signal, of course. But the problem is that we have also parts that contribute to decrease the signal, such like the absorber restriction coefficient and the absorber concentration. So the equilibrium between these two parts causes the amount of the exit radiation. And it must take care carefully, because if this is very strong, it means that we can have photodegradation. How we can avoid photodegradation? We have explained it before. In a microscopic Raman setup, it's not easy to avoid photodegradation because we have a small spot of few microns, and the collection optic makes that the confocal system concentrate the whole beam in a small amount, and therefore a photodegradation may occur. On a macroscopic Raman setup is more easy, also because we have a higher focal length, and we can oscillate the sample order, for example, to avoid photodegradation. We can move the sample order and to make measurements in an homogeneous sample, avoiding photodegradation. How we can avoid? We can, for example, this is what we use in our system. We have the laser source, we have the macro system, and we have sample oscillation and sample rotation. We are in a backscattering configuration, and the focus spot is around 50-100 microns, and therefore a photodegradation could be avoided. Now let's talk about selectivity of the excitation, as told before. How we can get this? With laser fixed wavelength sources, basically, by tuning with tunable wavelength source caused by synchrotron radiation or OPA laser, which warrants a better selectivity of the excitation source. How many minutes? Okay. Okay, let's talk now about the possible setup. Typically, what we have is a certain number of excitations. We have two. Basically, in our setup, we can use a triple grating of a simple grating system. In the first one, we can have a better resolution. In the second one, we have a smaller resolution. And we have basically broadband radiation coming from synchrotron that is monochromatized and then sent to the sample again. Now, let's talk about a real case of how to exploiting tunable source to study the DNA and RNA structural conformation. The absorption aspect of DNA is done in this way. which causes the absorption spectrum of DNA? Basically, the absorption is due to the presence of the nitrogenous bases. Each nitrogenous basis behave in a different way, and basically what we have is that if we exploit incident energies, place it in a different position in the energy scale, we can potentially have different effects. What we have is basically the DNA absorption spectrum, and means the electronic transition are due to the presence of nitrogenous bases, specifically the adenine, cytosine, guanine, and thymine that have a different absorption spectrum. So if we go to the maximum position of this absorption, we see that the maximum are at different position in energy. And what will occur if we get Raman spectrum exciting in different way? It occurs that basically if we go, for example, 272 nanometer and 260 nanometer, we have this kind of spectra. This is the DNA of salmon sperm DNA, standard one, which has a different line shape from the one collected at 260. This peak is lower, as well as the line shape of this peak is lower. Because this peak belongs to the thymine, this peak belongs to the guanine, more or less, and therefore the ratio between these two peaks changes, because in a case we are more, the guanine spectrum intensities is higher than the thymine one, increases, and in these cases the line shape changes. And if we go at 250 is the best condition to see guanine, for example, and of course in this case we have that the guanine contribution is the highest one. Also the other contribution are present, but the green one belonging to the guanine is predominant. Basically, this line shape is more or less equal, apart from the relative intensity of the peaks. But it occurs, for example, that when we go here at 228 nanometer, the spectral situation is completely different. Because in this case, we have the minimum in the absorption from all the nucleotides, apart from cytosine, which is a little bit increased in this case. And in fact, cytosine here is the predominant component, while in the previous one, cytosine was negligible, more or less. Cytosine is here, not so high. Cytosine is here, and cytosine is here, with a very high contribution. Why is so important, cytosine? Because it is proved that methylation of cytosine is indicative of the presence of cancer cells, for example. And therefore, a possible way to monitor the presence of cancer cell is to monitor the methylation of cytosine inside the DNA. To understand if it's possible to do it, we have done a test with DNA extracted from genomic DNA of hypomethylated cells, the green one, and hypermethylated cells. In this case, the spectra are completely different, proving that actually with exploiting synchronous radiation, it will be potentially possible to have a better selectivity from the Raman spectra between DNA of methylated cells and DNA of not methylated cells. But now, let's see how, for example, to get suitable Raman spectra of DNA, because the previous cases comes from salmon DNA, apart from this, which is a standard DNA, more or less, very used in scientific literature to make tests as a standard. But in the case of genomic DNA, we need to extract DNA from cells. And we should we must be sure that the spectrum is not contaminated. Because the problem is this one. The standard extraction protocols of DNA from cells are suitable to get the DNA for biological purposes. But we know that in Raman spectroscopy, even a small amount of traces of some chemicals that are particularly reactive in DNA in DNA cross-section may introduce in the spectra artifacts. For example, now, if we extract DNA from a QIAGEN commercial protocol from B16 DNA cells, we found a spectra that is completely different from some commercial salmon DNA. Why? Because we have the presence of basically two contaminants, ethanol and guanidinium hydrochloride that comes from the QIAGEN protocol. While the percentage of ethanol is relatively high, the presence of guanidinium hydrochloride is very small in solution, but since this molecule has a very high cross-section, it causes that DNA spectrum is contaminated. Therefore, we proceeded to improve to tailor the condition of extraction from the QIAGEN protocol to get a better extraction, and we worked in this way to get a tailored extraction that generates a spectrum very similar to one of the salmon tested commercial DNA, with the only presence of this mole belonging to guanidinium hydrochloride. But this part is substantially free from guanidinium hydrochloride and therefore can be used for further analysis, for example. And in this way, we have improved the spectral quality. For RNA, we've done the same things, but in this case, the problem is not the presence of contaminants. In this case, we've used the Trisol protocol, extraction protocol commercial, but the problem is the fluorescence due to the blank solution of the extraction protocol. In this case, instead of using a polynomial degree or something like this, we have collected a blank solution. So we have applied the protocol without cells. We get the final solution, we measured the final solution, and we found that this acquired spectra matches perfectly with our Raman spectra acquired on RNA solution. By subtracting, we found a spectra which recalls more or less the spectra of a DNA and therefore could be suitable for data analysis. We employ this protocol to extract RNA from spermatozoal cells, for example, and in this case we found an interesting thing that, as we know, we should use inhibition to avoid RNA degradation. Our question is how the presence of inhibitor may affect the Raman spectra. In this case, the response is that the presence of inhibitor warrants measurements on the hour time scale, avoiding the degradation of RNAs. And in this case, we proved also that the quality of spectra is quite high. In this case, we have extracted from RNA from standard cells. We have extracted RNA for a certain number of patients. In this case, the quality is high. In this case, the quality is not so high. Even we can go to very diluted RNA concentration because of the presence of proteins. Because if we subtract the contribution of RNA from the total spectrum, in the worst case, we found peaks that could be associated to the protein contributions. And so we found that the spermatozoa extracted cells are affected by protein contaminants. We should find a way to remove it. Thank you for the patience. So now I would like to show also these other experiments which can be done with the UV Raman and the interaction between SARS-CoV-2 G quadruplex RNA and drugs. The aim of this experiment was the identification of therapeutic molecules able to bind and stabilize SARS-CoV-2 G4 forming RNA, because in this case we can inhibit in some way the RNA and block in some way the viruses. And we should provide a plausible mechanism of activation of such molecule at the molecular level, because we would like to understand how these molecules interact with the G-quadruplex RNA. First, we have done a screening between a virtual screening between G-quadruplex and a huge amount of drugs approved by FDA, and we restrict our attention to 3 molecules that shows the best interaction with the G-quadruplex: quercetin, netazoline, and pranoclast. So now that we have obtained these results, how we can test the interaction between these and G-quadruplex? By means of UV resonant Raman scattering. And we have done this. Let's start with netazoline, this drug. First, here we have the complex, we have mixed netazoline with RNA, and here we have the complex spectrum. Then what we have collected the spectrum of the single RNA and the netazoline. Then what we have done, we found the correct coefficient to apply to RNA and netazoline in order to sum these 2 contributions and to give as close as possible to this curved line shape by minimizing this chi-square. Basically, we found the coefficient that give this curve more close possible to the sum curve. After that, in the case we have neither interaction, we should have that this different spectrum is exactly equal to 0. Of course, the spectra is not equal to 0 because the difference give information about the amount of interaction and where the interaction occurs, which is the most important things. Because here we can see different spectrum. Some differences are related to adenine peaks, and some differences are related to netazoline peaks. Basically, we have simulated the netazoline spectrum in order to recognize which part of the spectrum are modified. And what we found is that this modification belongs to this peak, which is attributed to this component, which is related to a specific portion of netazoline. Therefore, we can deduce that interaction occurs with this portion of netazoline. The same experiment has been done with quercetin, identified the portion of quercetin that interacts with the portion, in this case, not the portion of quercetin, because the quercetin contribution is very low, but we can identify the portion of RNA that interacts with quercetin. As with the pranoclast, we do the same things. Actually, the pranoclast has the lowest different spectral contribution, and the evidence is also the lowest interaction, as proved also from differential scanning calorimetric experiment, as well as circular decreasing experiments. Another thing that we can do with UV resonant Raman is to monitor the lysosome fibrils formation. Why? Because let's remember the complementarity between FTIR spectra and Raman spectra. In the FTIR, we see that the spectrum of fibrils and freshly made lysosome is basically characterized by 2 peaks, which are amide 1 and amide 2, for the people that knows very well the FTIR. And here we have differences. These differences are related to the modification of the secondary structure. But how we can monitor the tertiary structure? The tertiary structure could be monitored by the UV resonant Raman spectra, because in the UV resonant Raman spectra, what we see is not the contribution of the whole protein, but only we see here only the contribution of the aromatic portion, as well as as well as tryptophan and tyrosine. The decreasing of this peak related to tryptophan, let us suppose that a change in the tertiary structure occurs. And the same things occurs when we put resveratrol into lysosine, to verify if there is the possibility to change the fibrils dimension. From the AFM measurement, we see that we can change the fibrils dimension by using resveratrol, and what occurs here is related to less change into the tertiary structure investigated by UV Resonant Raman. Therefore, UV Resonant Raman is demonstrated to be a valid tool to monitor modification into the protein secondary structure. Paper is in preparation, the slide is old, but we have published the paper. If someone is interested, please ask me and I will provide all the information. Another thing that this is a relatively old experiment. What we have done is the analysis of graphene ion implanted into neuronal growth, and we have demonstrated that the possibility of we have seen a shift in the maximum peak in the graphene G bands, indicating the ion implanting in graphene layer, because my colleagues recognize a difference in the growth of this neuronal pattern. They suspect that it could be caused by the ion implanting in graphene layer, but was not able to demonstrate. By using the high resolution, look at this scale. We are at a resolution of a spectral resolution of 1 inverse centimeter, real measured at the resolution 1 inverse centimeter. We can monitor shift in peak position as well as in the peak line shape, demonstrating this interaction. Then we have used, just to pass to other topics, we have used UV Resonant Raman scatter to recognize the plastic fragments in jellyfish, identify the spectrum of jellyfish in high density polyethylene. Could these measurements done also with the visible Raman? No, because in this case we have a dirty sample, and the samples and the measurements of conventional Raman was affected by a huge amount of fluorescence, not obtained by employing UV radiation. Then another small experiment of gas interaction. By exploiting the fact that we don't have fluorescence background, we were able to study the interaction between caffeine into cyclodextrin-based nanosponges. And we are able to see that this spectra are flat. We don't remove anything here. This is not flat, the background, because we have removed a certain kind of fluorescence background or something else, but this is the real data, because in ultraviolet we can avoid the presence of fluorescence background. Therefore, we were able to subtract the caffeine contribution in a very careful way, and then to analyze the peak, to find these are Lorentzian peak, basically, to find from the width of the peak information about the dephasing time, and therefore to study the swelling of caffeine into cyclodextrin nanosponges. Then last example, then I close, because no, I can do also a few minutes. I have to finish. We use also UV Raman to exploit the certain smart materials, such like as tannin-based rigid forms, that are innovative, sustainable materials made on inexpensive organic ingredients that can be used as substitute of plastic for many purposes. Tannin basis formation is an acid catalyzed polymerization that involves a self-blowing reaction in which the foam raises up, introducing a preferential direction of blowing. We have these final products. And now, what is done? Basically, we put two main ingredients, mimosa tannin, we put furfuryl alcohol, we make catalysis, we use blowing gas and surfactant, diethylene glycol in a small amount, and we have finally a heteropolymeric structure. The problem is that the macroscopic properties of these forms are related to its heteropolymeric structure. How we can determine the heteropolymeric structure of a such complex system? By employing more than one spectroscopy. First of all, we analyzed the behavior of polyfurfuryl alcohol, because furfuryl alcohol have polymerization inside here. And then we study separately polyfurfuryl alcohol to understand how does it behave. We can have black viscous liquid polyfurfuryl alcohol or thermosetted one. Basically, we expect that in the previous sample, we have a combination of both, but we don't know if the composition in the heteropolymeric form is similar to the black viscous or to the thermosetted one. We don't know it. We studied it separately. The problem is that when we study the FTIR spectrum, this is the FTIR spectrum are very similar, apart the small differences, and are not able to infer about this variation. But if we employ the UV Raman, in UV Raman, we recognize that we have strong differences with these two samples, passing from visible to the ultraviolet. And in particular, in the ultraviolet, we found that all the peak disappears, and we have only these two peaks that are more intense in the thermoset than in viscous, the ratio between this peak, that make information of the polymerization. Therefore, we can do measurement of tannin extracted 266 to recognize the flavonoid contribution. These two peaks that belongs to the flavonoid ring breadth. This is the spectrum in the ultraviolet resonant Raman of tannin of pure tannins, and here we can monitor the amount of polyfurfuryl alcohol inside several kinds of tannin extracts. And therefore, we can use UV resonant Raman to monitor how many chains of polyfurfuryl alcohol we have inside these forms. The last example about organic semiconductor. This is an organic semiconductor. It is used to make devices such like solar cells, but to study the chemical conformation of this film, we should have a way to investigate the thin film, because in the bulk, we know the structure also from XRD, but from thin films, in particular on the surface, we don't have any way to understand how these molecules behave on the surface from the chemical point of view. What we can do? We exploit resonant Raman. The penetration depth, this is the absorption spectrum of our film. The penetration depth depends from the excitation. When we go at 633 nanometer, 530 nanometer, we have a higher penetration depth. But at 260 nanometer, we have a very small penetration depth, and therefore we are analyzing only the surface layer of these films. And this is also reflected from the spectrum, because in the spectrum, here at the higher part, we can see difference in the spectrum basically related to the resonance. But if we go to lower wave numbers, these are simulations that we have done to understand which kind of vibration we are analyzing, we see that this peak decreases in intensity, passing from the visible to the ultraviolet. And in particular, we see that at 266, at these two excitation wavelengths, we cannot investigate surfaces, because basically we can study the powder, but the film appears with strong fluorescence. At 266 we can, and we see difference in the spectra between film and powder, not at all the normal modes, but only near specific normal modes that are modified in position. Which are these normal modes? The normal modes that contains also out of plane vibration. Therefore, we have a way to investigate what occurs on the surface and to distinguish between surface status and not surface status, and investigate how the films behave